Okay, good morning, everyone, and welcome. It's a pleasure to continue the distinguished lectures of the Josep Carreras Leukaemia Research Institute. Today we'll have a hybrid format, so we have the online presentation and the presential lecture. And in this regard, it's a great pleasure to have with us Professor Iannis Aifantis. A real pleasure, and I would like to thank him the effort to come to Barcelona in always difficult times. Professor Aifantis is widely known for his expertise in the fields of hematopoiesis and leukemia. He's the professor and chair of the Department of Pathology at New York University, Grossman School of Medicine. His training came originally from the University of Crete in Greece, where he earned his PhD in the University of Paris in France. He completed his postdoctoral training at Harvard University, at Dana-Farber, both in PhD and postdoc, studying T-cell signaling and differentiation. And later he moved as independent investigator to the University of Chicago until 2006, that he joined NYU. Through his career, he had received numerous awards, including the Vilcek Award for Creative Promise, the McCulloch-Till Award for the International Society for Hematology and Stem Cell Biology, and the Frederick Alt Award for the new discoveries in immunology. He's also an early career investigator. In 2009, he was an early career investigator by the Howard Hughes Medical Institute. He is, of course, one of the leaders in the fields of hematopoiesis and leukemia, in many different areas, from protein stability to epigenetic regulation, tumor microenvironment, and it looks now non-coding RNA modifications, trna biology. He's widely known for his contributions to the understanding of how it's initiated and progressed different leukemias, among them acute lymphoid and myeloid leukemia. So the title of his talk is going to be Malign tRNA biogenesis and viability in T-cell acute lymphoblastic leukemia pathogenesis.
Thank you so much, Manel, for the invitation and having me here, and everybody else for being here. It is my actually first lecture, second, because yesterday I was at the IRB downtown, and I'm very happy about it. Let me see. So my lab is doing different things, as Manel said, and these are some of kind of the main foci at this point of what we're doing in the lab. Yesterday, I told people a lot about our single cell work in acute myeloid leukemia and the putative role of inflammation in transiting from clonal hematopoiesis to AML. But today I chose to present something that is relatively new for us. It's probably the first time that I present it, so bear with me, it's not going to be perfect. But it's a very nice story that actually it's coming out, I think, next month for publication. And it's a story on TLL, not AML. I know that some of you work on ALL and specifically TLL. TLL has been kind of the first lab of my lab, and then slowly moved to the myeloid side. But TLL and specifically ALL in general is a success story, basically, for treatment, usually chemo-based treatments that basically manage to cure a lot of children with acute lymphoblastic leukemia. However, adults are not doing that well, and children that basically relapse, we don't really have means, especially in TLL. BLL is a little bit better because of the CAR T-cell treatments at this point. Several labs, and in this case, I'm giving you an example from Adolfo Ferrando's lab, have studied oncogenic signaling TLL, and for years we have studied NOT signaling and induction of MYC downstream and such things. And I think one thing that we have learned is that most likely we're not going to target NOT signaling itself, but you should go downstream and target things that are induced by Notch and targets of Notch. Again, from Adolfo's lab, it was a realization. Actually, this is work done by Teresa Palomero. It was a realization. Basically, Notch can directly impinge on translation, ribosome biogenesis, and in this case, tRNA synthesis. Something that made us really interested in this idea that oncogenic signals can basically control translation directly. And this is not something totally new. People in the solid tumor field have studied translation. They used drugs that can target translation at different stages, and more recently focused on profiling tRNAs and even measuring charging of tRNAs with specific amino acids and their effects on progression of the tumor. So the work that I'm going to present you was done by a postdoc in my lab, Raja Thandapani, who was focusing on that for quite some time. And it started relatively simple. We never thought of tRNAs before, I don't know, 3 or 4 years ago. But when basically Raja was profiling genes that change their expression in TLR, and you can see some of them, and we focus specifically on translation-specific genes, we have seen some interesting things popping up, including VARS, which is basically a synthetase for valine, placing valine on tRNAs and inducing progression of translation. So this is kind of your classical textbook view of tRNA with the anti-coding and the codon on the RNA, as you can see here. So the next thing that we have done is that we have taken a better look at dynamic of gene expression and RNA Pol2 progression. In this case, we're doing gross sequencing. Gross sequencing after basically wash off of gamma secretase inhibitors to basically calculate genes that are directly controlled by Notch. So VARS has been there again. And I don't know if I have it in this presentation, but that VARS is actually a direct target of Notch. It is bound and directly control its transcription by Notch1. And the same thing, an interesting tRNA, which is coded by a lot of different fragments of the genome, that is basically a valine TAC tRNA, and keep it in mind because we're going to mention it a little bit later. So the next thing that we have done is, of course, RNA sequencing. We wanted to see how many of these genes can be modified also at the level of the RNA. VARS is, so as I told you, it's a direct target. But if you look now at tRNAs, you can see the valine tRNAs being controlled in TLL, including this valine TAC. And not only they're changing at RNA sequencing level, but the charging of the tRNAs basically drops once you inhibit Notch activation. And this is very similar to what you see if you just take valine away from your medium. The next thing that Raja has done is basically run CRISPR screens, and we decided not to do whole genome screens, but do screens that basically target directly proteins and genes that control translation and tRNA biogenesis. And these are two different TLL cell lines, and you see again the valine synthetase VARS being one of the strongest genes that pop up in these particular screens. This is another factor that people, especially in solid tumors, have been in the field of translation, have been focused a lot. So what happens now if you just take primary TLL samples and you look at expression of these genes? Basically, what you see is that VARS is, as we expected, regulated and highly expressed in TLL samples. This is maybe a better comparison, comparing actually human T-cell progenitors and not mature T-cells, and you see again VARS expression being much higher in primary cells from TLL patients. As I've told you before, VARS is directly bound by Notch. This is the TSS of the gene with H3K4 trimethyl. This is what I've shown you before with a gross sequencing, that you switch off inhibition of Notch and the gene comes back. And if you block it by gamma secretase inhibitors, you basically drop the expression of VARS and the tRNA that I've discussed before. The next thing that we have done is a little bit more sophisticated, and it's basically tRNA profiling. We're using a protocol by the Tavazoy lab at the Rockefeller, as published a few years ago, that basically includes small RNA extraction, deacylation, pull down with biotinylation, use of specific probes that cover all the tRNAs, and then basically profiling of tRNAs. And if you now compare TLR samples with, in this case, normal T cells, you see again the valine tRNAs being really overexpressed in TLL, including this valine TSC tRNA. So then, how can we target tRNAs? They are tricky because they're produced by a lot of genes. They look like each other. So there's noise in the system. So we decided to do it two ways. I don't know which one I have here. I think I have the Cas9 here. Here we're having, so we've done it two ways. One is Cas9, which targets basically exactly the anticoding loop here by creating indels. And the other thing, and I'm not sure if I have it, I think I have it a little bit later, we're using a different Cas13D, which targets specifically RNAs. So we can target the tRNA at a different basically area, and doesn't have to be in a conserved loop structure. So, but this is Cas9. We target the anticodon loop, we create indels, and as you see here, our TLL lines basically die pretty fast with the same kinetics as we target VARS, with the same kinetics that they die when we actually take valine away from the system. So now that I said that, of course, the next question is, can we alter valine levels by just providing a food to our mice, in this case, that doesn't have valine or has very low levels of valine? And this is something that is feasible, has been done before. And actually, it has been done in a more severe case. So children that have certain metabolic, first of all, as you all know, asparaginase has been used in leukemia to target asparagine for quite some time. But children that have specific types of metabolic diseases, in this case, one example, methylmalonic acidemia, they're actually taking medical foods that contain only trace amounts of specific amino acids. So in this case, we don't have to take all 4 of them. We can just take one of them out of the food. And the question is, you know, what happens? That has been tested before, in this case with methionine in animals and solid tumors. So we decided to test it in leukemia. And the results were actually really breathtaking for us. And again, you don't have to be a leukemia expert to realize that this is basically splenomegaly that you're getting in these animals if you have normal levels of valine. And this is what happens when you just take valine out. We believe it's because of apoptosis. We see an exin-5 cascade activation and such. This is the peripheral blood of these animals. You take out valine for a couple of weeks, and they basically drop. I think this is 3 weeks, yes, 3 weeks after you deprive valine from the system. We have done it with human pdxs, really same results, basically drop in the levels of leukemia cells in peripheral blood, as you can see here too, and PDX animals live much longer. However, you cannot take valine out of your system for a long time. For obvious reason, muscle wasting, effects on your hematopoietic system. So there must be a balance there. So what we decided, so first of all, before I get into that, this is a little bit of a comparison with other amino acids. You take out asparagine, not much happens. You take out lysine, maybe something happens, but valine is the one that is actually much more strong and gives you much more significant results in this system. However, as I told you, you cannot, for prolonged periods of time, take out valine because it's detrimental hematopoiesis. Not really hematopoiesis. This mice actually get eventually, if you leave them for a long time, muscle wasting. So we decided we can do different things. First of all, you can combine it with different drugs. Second, you can play with concentrations, and I'll just start with that. So what happens if you take out valine, the cancer goes away, and then you add valine back, and as you see, and that was an amazing result, the cancer comes back actually very efficiently. So what we decided to do is we play with concentrations, and instead of taking valine totally out of the system, we use different concentrations of the system, and we can find that there are certain concentrations that can give us a pretty significant prolongation of a lifespan in these PDX models, but at the same time retain almost normal hematopoiesis and weight and muscle tone in these animals. And I won't tire you basically with this concentration. This is what you get when you, in body weight, when you take valine totally. This is what you get when you're in this intermediate concentration of 0.8 Gs per liter in this animal model. So let's go a little bit on the mechanism. So how is valine functioning in this system? So the first thing that we have actually done is we have used ribosome profiling to basically take a better look at translation and see which are basically the RNAs that are specifically affected. Of course, we expected that this would be RNAs that are heavy in codons with valine, so heavy in valine. And so these are basically ribosome profiling experiments. You can take valine totally out or in different concentrations. We can see specifically RNAs that this is whole genome, that's why it's so noisy. I'll show you better examples later that are affected, that basically have valine codons. And if you now look at genes that are specifically affected, you can see this 32 in the middle, but there are more than that if you just take two concentrations here. You can see a lot of really interesting genes, but the thing that really pops out for us, and it will take me to actually the title of my talk and the next part of the talk, is that the genes that are affected, they're all in the mitochondria. And that was a huge surprise for us. We did not really expect it. It was a very interesting surprise because as I'm going to tell you, we do a lot of mitochondrial work in different systems. But basically, it was mitochondria and electron transport chain, especially the respiratory chain complex 1. And then if you go whole genome and you do some proteomic analysis here, you actually see, that is complex 1 key proteins that are basically very heavy on valine codons and are particularly affected in these ribosome experiments that we're doing. And to better prove that, we have taken two mitochondrial genes, one that has 12% valine, so pretty high, and the other has 1% valine. And the one that has 12% valine, in this case, we made reporters actually in cells, and we can see if we take valine out, the one that has 12% is much more efficiently targeted as you would expect than the one that has only 1%. So big surprise and really exciting for us to see that valine, for some reason, preferentially, of course it targets other things, but preferentially targets the mitochondria. So for the ones like me that are not thinking of basically mitochondrial complex 1 chain, a little bit of information. So complex 1 on the surface of the mitochondria is basically the first enzyme of the respiratory chain. It oxidizes NADH and basically reduces ubiquinol to ubiquinol. And it's a large complex of 40 plus proteins, and basically is absolutely important for metabolic abilities of every cell. So now what you can see, if you actually use blotting to identify the way that the complex is formed, you see if you take out valine, you see basically the complex is specifically not formed correctly, suggesting that valine deprivation leads to basically defects in the assembly of the complex. We wanted to see how specific is that. Is it basically the valine pathway itself? So what we've done, and I mentioned that before, we targeted not valine here, but we targeted specifically VARS, the synthetase, and we've done it using Cas13D that we're using a lot in the lab recently. So we're targeting Cas13D, targets the RNA itself and not the DNA, and you can see that the cells are particularly affected. If we go early on and we repeat basically this Western blotting analysis, you see specifically disassembly of complex 1 formation in these cells with different guide RNA specific VARs. So it's VARs, it's valine codons, and specifically utilization of this in the respiratory complex 1 complex. Of course, that translates in metabolic defects. So valine deprivation leads to mitochondrial respiration defects. This is your classical seahorse assay, basal respiration. You uncouple your mitochondria and you get your maximal respiration. And we can see differences. So zero valine is this pink one, and the green is the control. We can see differences both in basal respiration and after XCCT uncoupling at maximal respiration rates. Pretty significant differences at that level. And not only that, but more downstream, we see basically reduced NAD, NADH ratios and increased mitochondrial ROS basically upon loss of valine in these cells. So to finish this story, all of these things happen basically on the inner membrane of the mitochondria, in what people call a cristae. Cristae is very well known because it controls, as you know, cytochrome C release using this OPA1 protein that basically keeps the cristae closed. And this is where you find on the cristae the respiratory complexes and the ATP synthetase complex at the same level. So people in the lab, and specifically Christina, who I'll tell you more about her work a little bit later, are mitochondria physiologists, and they're thinking more of the mitochondria. So the question is, what happens to the actual mitochondria membrane when you take out valine? And basically, so this is how mitochondria look. This is electron microscopy. This is normal valine, and this is low levels of valine. This is normal cells, and this is when you knock down VARs specifically. So basically, by doing electron microscopy and structural studies, we can see that valine restriction and VARs knockdown is inducing changes in the mitochondria, specifically changing the actual width of the cristae in the mitochondria. So the width of the cristae is increasing, and what we call the disorganization of the cristae is increasing. So the last thing that we have done was, we said, look, valine deprivation is not a drug, probably is not going to work ever as a monotherapy. So can we actually propose that can work as a combination with a different drug? to identify drugs, or in this case, genes that actually can cooperate with valine deprivation, we decided to run, in this case, whole genome CRISPR screens. And we have run screens both in normal levels of valine and when we deprive the cells from valine. And basically, when you do that, you get this classical sigmoid curve, as you expect. And we should have done it actually first and get into the mitochondria, because as you see, almost all proteins here are mitochondrial proteins. You see them all here, but on that side, which is basically the sensitizers, the negatively selected cells, you see, of course, VARs, and that made us believe that, you know, our screen is actually correct and works. You see the branch amino acid transporters, the two SLCs, and that's really important. You see again some mitochondria metabolism proteins, this PTCD1, which is important for mitochondrial translation, the NAM PT, which is basically part of an NAD salvage pathway. The cells are trying to salvage NAD when we take out valine. And another and well-known protein is, of course, Bcl II. So I'll show you a couple of data with SLCs and Bcl II for very, very obvious reasons. So what are these SLCs? And specifically the 7A5 is basically, it's a large neutral amino acid transporter. It basically supplies cells with essential amino acids. This is how it looks. And basically when you target it, and you target it specifically with deprivation of valine, you see this synergistic effects of killing of TLR cells. In this case, we're not using a guide RNA, we're using a compound that specifically targets this particular transporter. The second protein that we are, as you will see very later, really excited about to target is obviously Bcl II. And that's because Bcl II, as I'll tell you a little bit later, has been targeted, of course, in leukemia, CLL and AML specifically. So if you target Bcl II with guide RNAs and you take out valine, you get this really strong synergistic effect. And if you add venetoclax in this case, you see the same synergistic effect. And that takes us back to work that people have done in TLL and in PLL, and suggest that basically you can use venetoclax or maybe another BH3 mimetic and combine it with low levels of valine in the food of these patients. So what I've told you in this part is that TRna deregulation is a critical adaptation in TLR pathogenesis, that Notch1 can directly control both VARs and specific tRNAs, that dietary valine deprivation reduces leukemic burden, that if you take out valine, you see specific effects on complex 1 assembly and function, that valine deprivation could be combined with BH3 mimetics. And that takes me to the second part of my talk, that drug response and resistance is coupled to organelle adaptation. And this is really important because I know that all of you know about mutations and adaptation of the cells of treatment by basically mutating. Manel and a lot of you are working on epigenetic adaptations, but what we propose here is that you can have another level of adaptation, and in this case, it's basically at the function of specific organelles, and mitochondria is one of this type of organelles that adapt to drug treatment. And Raja's work is coming out next month, I believe. We are very happy about it. So this first part, most of the work done by Raja, Christina trained as a mitochondrial physiologist with my friend Lucas Corano, who is now in Padova, and we could not do all these things without the help with Aris, who gives us always bioinformatics support, Thalis, who is a metabolomics guy at NYU, and Sohail, who helped us a lot with tRNA. So in the second part, which is probably shorter from the first one, I will just tell you why we are thinking that much about organelle adaptations and why we think that mitochondria are so important in adaptation to drug treatment. So as you know better than me, most likely, mitochondria is an organelle that does a lot of things. It controls energy production, it controls apoptosis, but of course, calcium homeostasis and availability, and it includes a lot of signaling pathways within the organelle. Mitochondria, they're not static. They change, they fuse, they elongate, they basically kill themselves with mitophagy. And that's an exciting way of basically responding to different extracellular stimuli. As I've told you before, the crystal of the mitochondria is the key component of the apoptotic machinery. OPA1 is the gatekeeper there that opens and closes the pore. And if you have an open pore, you get cytochrome C out, and that basically leads to crystal remodeling and anti-apoptotic protein or proptotic protein oligomerization and induction of apoptotic death. And this is work that was done by a lot of people. Luca, when he was a postdoc with Stan Korsmeyer, and later work from his and a lot of other labs. Why we're so excited about that? Because of drugs, and I know that a lot of the patients that you see here, both for CLL and AML, actually treated with pdxs mimetics, and specifically venetoclax, but other drugs are out there, MCL1 inhibitors and combinations of them. Venetoclax has been approved first for CLL and then for AML in combination with 5-ASA or decitabine, or basically low dose cytarabine. So what we wanted to do in the lab all these years is basically ask this very simple question, coming from more of a basic biology component and side, is that which are the underlying mechanisms of resistance to venetoclax? So again, this is work that Christina and Shufenk have done in the lab for years. The first part is published, and I'll give you some of the data that we are putting together now as a follow-up. And again, we started with CRISPR screens, very simple CRISPR screen. Let's get AML cells and basically do whole genome screens with or without venetoclax. And these are different concentrations of venetoclax here. And when you do these screens, you see first all your obvious culprits basically here. You see BAX, you see NOXA, you see P53, and on the side, other side, MCL1 that tries to go up, MDM2 that controls P53, etc., etc. And also other things that got our attention is this mitochondrial gene that's called CLIPPy-B, and I'll show you a little bit more about it. It's a chaperoning on the surface of the membrane of the mitochondria. So what Christina and Shufenk have done now, and I'm just giving you a slide on two of that, we have basically rescreened by doing venetoclax with 5-ASA, 5-ASA itself, MCL1 inhibitors, venetoclax with MCL1, all potential combinations. And you see very similar data, BAX, 53, BAC, NOXA on that side. Interesting genes like FLT3 on the other side, CDK2, and another, actually the clinical trials combining venetoclax with FLT3 or CDK inhibitors. So we are basically studying some of these combinations in the lab at this point. Also, together with Marina Konopleva and colleagues at Michael Andreev and colleagues at MD Anderson, we have actually sequenced actual patient samples coming from patients that either they're in durable remission after venetoclax 5-ASA, or they relapse, or they're refractory. And basically, these data are very similar with what we found in our CRISPR screens. P53, FLT3, RAS pathways, some of the pathways that are involved in induction of resistance in these patients and in these cells. So what is out there, apart from your classical P53 and FLT3 and maybe the RAS pathway? So what is out there, and that connects to my previous part of the talk, is basically mitochondria and mitochondrial proteins. If you look at basically genes that are depleted in the screen, so genes basically that when you knock them down, they cooperate with venetoclax and BSC mimetics, are basically a lot of mitochondrial proteins. And so the first thing that we have done is that, so what happens to the mitochondria when you add venetoclax? Basically, we have seen that the venetoclax very efficiently disrupts CRISPR ultrastructure. You see this nice CRISPR, very tight ones, and you see these disorganized organelles when you add venetoclax. I've shown you before that one of the main components that control cytochrome C release is OPA1. So what was really exciting to see that when you add venetoclax, you basically cleave proteolytically OPA1 very efficiently. And you get only the non-cleave form of the protein. So then what we've done is that we have decided to create, not only do screens, but create cells that are resistant to BSC mimetics. And we've done it two ways. We have made cell lines, AML cell lines, that we have treated basically for months and months with venetoclax and MCL1 inhibitors and such, and we made them resistant, or we got cells from actual patients that relapse from treatment. So basically what we've seen in these cells, and you can see it here, is that venetoclax resistance is coupled to OPA1 overexpression. I think it's somewhere here on my slide. So look at the overexpression of OPA1. Huge amounts of OPA1 overexpressed in resistant cells. And now the mitochondrial crystal is much more tight because they have a lot more OPA1 to keep it closed. So targeting of proteins involved in the maintenance of mitochondria could act synergistically with venetoclax treatment. And the other protein that, as I've told you, we are particularly interested, apart from OPA1, is this CLIP-B. CLIP-B is an enzyme on the surface of the mitochondria. And the interesting thing is actually it's overexpressed in AML patients. And as I'm going to show you, it's even more overexpressed in AML patients that are resistant to venetoclax treatment. So I'll actually go to the next one, it's probably better. So this is what happens now. AML xenografted, resistant to venetoclax. We see huge amounts of overexpression of KPP and OPA1 in these cells. So then we said, what happens basically if you knock down CLPB? So if you knock down CLPB, you basically very efficiently reduce the IC50 for venetoclax, suggesting that if we had a drug to target OPA1, sorry, CLPB, we could actually combine it with venetoclax treatment. We have done similar studies in vivo, and we can see the same thing, that basically CLPB knockdown is synergizing with venetoclax very efficiently. The problem in this case, and before I go to the problem, I'll show you one more slide. So what happened in cells that are actually P53 mutants and they are refractory, let's say, to venetoclax treatment? They do respond to CLPB knockdown pretty efficiently. So if we had this drug, we would actually combine it. The problem is that we don't have a drug for CLPB at this point. There have been some compounds targeting bacterial CLPB-like proteins, but not actually a mammalian one. So we decided to find ways around it, and we tried to get that by first studying the function of this protein. I've told you it's a member of this ATPase superfamily. It hydrolyzes ATP. It's on the surface of the mitochondria, and it's involved in a lot of mitochondrial functions. And by doing ablation of CLPB, we could very easily see that basically if you lose CLPB, you impair ultrastructure of mitochondria itself the same way that you do when you lose OPA1 or you add venetoclax. So what happened to OPA1 itself when you get rid of CLPB? You cleave it. And that was actually a really interesting result for us. This is your controls. You see the uncleaved one here, your cleaved one here, and you see the loss of cleaved OPA1 when you knock down CLPB, suggesting to us that these two proteins could actually function together. And additional biochemical work that I'm not going to tell you, it's published, show that you get a very nice interaction between CLPB and OPA1 on the membrane. So that gave us the idea, well, we cannot target CLPB because there is no drug to target CLPB. Can we actually target OPA1? Exactly that. Can we drag the CLPB-OPA1 interaction? And we were lucky because Lucas Corano and his lab have published last year at Cell Metabolism a paper in which they have described this, the first in-class inhibitor for mammalian and human OPA1. So we sat down with Luca and we used this particular drug. And I'll show you a little bit of data. First, some guide RNA data to just make sure that we are kosher here and the drug is not doing anything weird. You knock down OPA1, you induce cell death very efficiently in AML lines, even lines that basically are mutant for p53. And now if you use the Scorano drug, that's the original publication, you see really impressive synergism in AML lines, even lines that are resistant for months and very high concentrations of venetoclax itself. So basically what I've told you in the second part is that mitochondria again is the organelles that they are really attractive for the study of resistance in cancer, obviously because of their involvement in metabolism and cell death. So I gave you a couple of examples, I think, today. In the second part, I mentioned to you about CLPB on OPA1. So CLPB loss sensitizes to venetoclax treatment by inducing remodeling of the crystal and mitochondrial stress response. And what we're doing right now is that we are using the compound from the Scorano lab in AML xenograft preclinical settings with or without FLT3 mutations and p53 mutations, and we try to see if we can actually take it from a preclinical side to a clinical trial that we started putting together at the cancer center at NYU. So the second part, the work was done by Christina and Shufeng. They're both postdocs in my lab. A lot of work from Evripidis, who is studying actually cell death close to us at Albert Einstein and the Bronx. Luca, I mentioned, Raul for a lot of clinical trial studies, and samples and clinical support by Marina and Michael Andreev at the MD Anderson Cancer Center. I'm going to stop here and get questions. Thank you all for being here.
Thank you. Thank you very much. Wonderful presentation. So before giving the word to the audience, just we're working also on tRNA modifications and tRNA defects and all that. Was the first part was a surprise for me and beautiful, very nice. Both to the first second part, there are type of drugs that maybe you try this. are the glycolytic inhibitors. Glycolysis inhibition in these cases, it works, it provides a different sensitivity in your models, or does not? Did you try it?
We haven't tried it. The prediction is that it will provide sensitivity just because of the actual mitochondrial function and the feed into the glycolysis pathway. We see glycolytic enzymes being upregulating, kind of like try to salvage the system. So so yes, we haven't tried it, but it's glycolytic inhibitors and also mitochondrial translational inhibitors. And I'm going back to this, sorry, to this screen that I've shown you before. In this case, you see the
and this the green, no? The green ones.
So yes, absolutely.
Thank you. For the questions you want, you can take out your mask to ask the question, to be clear that can be understood.
Hi, thank you for the talk. That was fantastic. So I was also thinking about the first part. If the tRNA for valine is the only thing that you require for the valine restriction phenotype. I mean, is it epistatic, or is there any function of valine that is beyond the tRNA and the protein synthesis?
So based on this targeting of the studies of this specific tRNA, the valine TAC, it's encoded by 5 different genes, basically. We believe that it's actually the tRNA itself that does it, and valine does not have kind of like non-tRNA function. So you can get the same phenotype taking out valine or taking out VARS or valine tRNA.
Iannis, thanks a lot for these 2 presentations. I also would like to follow up on the valine. Is there any evolutionary explanation why complex 1 should be hypersensitive to valine concentrations?
That's a great question. I don't know. I was talking with somebody. Will have to look at evolution of species and I guess utilization of these tRNAs. I don't know if as a species we kind of have evolved what we eat, basically. I'm trying to think of meat and switch of the diets of humans during the years. Cool question. I have no idea.
Thank you, Iannis. I was wondering about the effect of venetoclax on the mitochondria crystal. Is this permanent? So, and what happens, or this is even not only venetoclax, also when you target the OPA1 and when you target the other proteins, do you think this takes to some other alterations on the mitochondria, or this is a permanent, that then they go to cell death?
They definitely go to cell death. I think that there is a time point. I think you can revert, basically, if you have shorter time points. We have actually seen that. You definitely go to specific alterations of the membrane if you keep the drug for a long time. And it's really interesting to see the cells that we made resistant after months of treatment and increasing the concentration. They really had like really tight, close crystal, suggesting that you get a very strong adaptation at the level of the crystal to the drug. We see very similar effects if you take out OPA1, of course, disorganization, and TPP.
Hi, Iannis. So a question related to Anna's question. So have you checked if the venetoclax effect on mitochondrial structure is directly linked to Bcl II interaction? Or in other words, do you think that this effect could be another way to work of venetoclax?
Yeah, I was discussing that with Tony Letai, who does this kind of work. I assume that it's directly linked to Bcl II. You could rescue your phenotype if you overexpress Bcl II or MCL1, but you can rescue the apoptosis phenotype. We never really looked at the ultrastructure of the mitochondria if you try to overexpress Bcl II at the same time that you. But I would assume that is with Bcl II related. But we've never, let's say, knocked down Bcl II and seen if there are similar effects. I would expect that there are similar effects.
Okay. And another question regarding your first part on the tRNAs. I have no idea. I'm not on the RNA field.
Me neither.
I am on the TLL field. But in addition to this kind of more RNA or different RNAs on the notch-induced TLL cells, but there is any structural difference on this tRNA versus non-leukemic tRNAs?
I don't think so. I mean, based on at least, we never looked at structure, but I mean, based on the tRNA profiling, I don't expect, I mean, you expect preferentially utilization or less utilization of tRNAs based on the subtype of, or the cell type that you have, but I don't expect any differences. I mean, the exciting thing with tRNAs is that they can be cleaved, and there's literature on cleavage of tRNAs and using fragments of tRNAs basically to perform functions that are not directly translation related, which is another story, and we haven't really profiled it at that level.
Thanks for the talk. So also going back to the Valin story, so does the alteration in the structure of mitochondria activate the inflammasome within those cells and they die by pyroptosis? And also following up, is different sensitivity to pyroptotic death, which makes some cells die first and then later on the predation strikes other cell types?
I mean, great questions. We have never looked at pyroptosis. We only looked at apoptosis just because of the very classical functions of the Bcl II family in this system. And also in our screens, we have not seen non-apoptotic related genes popping up. So what was the first one? Sorry, I forgot about it.
Yeah, if they die by pyroptosis, and then if there are different sensitivity and different cells to pyroptotic death. And
great question. We assume that there is not just because of what we've seen in our screen, but we've never actually looked at that.
Okay, we should finish here. Thank you very much.